You can probably go ahead and start whenever, though.
Okay, great. All right, thanks so much for having me. Hello, everyone. As everyone's filtering in, I guess I'll get started. My name is Ryan Weiss, and I'm an assistant professor at the University of Georgia. And my lab is located at the Complex Carbohydrate Research Center at UGA, specifically right here. I recently moved here from the University of California, San Diego, and started my own lab as an assistant professor at the CCRC in January of this year. And yeah, I'm really excited to be part of this course that is offered, I believe, every single summer. And so I know you've all been learning a lot about GAGs already and how we structurally analyze them. But I guess the focus and the purpose of my talk today is to kind of give a little bit of a broader background in terms of the biology and the structure and function of GAGs, and also their use in biomedicine as well. So if you have any questions, I think the best thing to do is to type them in the chat, and I guess we will, at the end, Stephanie will help and we'll address the questions at the end. But of course, if you have something that makes no sense along the way and you want to unmute and ask the question, you're welcome to do that as well. As I'm sure that most of you are aware at this point, if you've taken these courses over the last couple of weeks, all cells are surrounded by glycans. So these complex carbohydrates are typically attached to glycolipids or glycoproteins on the surface of all of our cells. And collectively, it's known as the glycocalyx that forms this protective coating on the outside of our cells. So this network of glycoproteins and glycolipids serves not only to protect the cell, but it's also a way that the cell is able to interact with its environment. And so you can imagine that anything that comes in that wants to interact with the cell, this is the first thing that it will interact with. So these glycans are very important for many important cellular processes that we'll talk a little bit more in depth later. And also these glycans are also a key structural feature of the basement membrane and connective tissue as well. And as you've kind of learned in other sections of this course, there are many different classes of glycans that coat the cell surface and are found in the extracellular matrix. And I'm not going to focus on a lot of these other different classes, but just wanted to show in the context of all different types of glycans that there are many different types, and they all are very important for different cellular functions. But today we're going to be focusing on glycosaminoglycans. So these are long linear polysaccharides that are found either attached to core proteins or they exist freely in the extracellular matrix. And what makes these unique is that they are typically all linear, and most of these GAGs, as we call them, can also be sulfated at different positions, which makes them highly negatively charged, which allows these GAGs to interact with many different factors and proteins outside the cell, on the cell surface, in the extracellular matrix. So I wanted to dive in a little bit more, and we're first going to talk about proteoglycans. So proteoglycans are the core proteins that these glycosaminoglycan polysaccharides are attached to. And proteoglycans can either exist on the cell surface in the form of transmembrane type proteins, such as the syndicans, or GPI anchored proteins known as glycans. And these GAG chains can also decorate many different types of extracellular matrix proteins as well. And all of these different proteins are involved in different important biological processes, and we're going to talk about the different types more in detail later. So we talked a little bit about the core protein itself, known as the proteoglycan, but next we're going to dive into the structure and function of the actual glycosaminoglycan chains themselves. So there's many different types of GAGs, and they are separated into these different classes depending on their sulfation pattern, as well as the type of monosaccharides that are made up of these polysaccharides. And so first I wanted to talk about the differences in structure as well as function of these different glycosaminoglycans. So the first one is called hyaluronan, or also known as hyaluronic acid, or HA. So hyaluronic acid is very unique compared to the other types of GAGs in that it is non-sulfated, and it is the only GAG that is biosynthesized right at the cell surface of the cell membrane. And then as it is biosynthesized, it is pumped outside of the cell into the extracellular space. And again, it is non-sulfated, but the hyaluronate chains are actually made up of a repeating disaccharide subunit, which includes glucuronic acid and N-acetylglucosamine. So as you probably gleaned from other courses in this training for Glycoscience class, we typically show these different sugars as these different shapes. And we just use that for ease in terms of, instead of drawing them as their more complex chemical structures. But each of these shapes just stands for the different types of sugars. So here I'm circling a glucuronic acid and N-acetylglucosamine disaccharide. And here, if you look at the actual chemical structure, this tells you a lot more about what's actually going on in terms of its structure. So these hyaluronic acid polysaccharides are negatively charged because it has this carboxylate group, but they are not non-sulfated. And interestingly, these hyaluronic acid chains can be very, very large, up to 7 million Daltons per chain. And they actually are the chief component of the extracellular matrix. Again, they're biosynthesized at the plasma membrane. We'll talk a little bit more about that later in detail. And HA is distributed widely through our connective tissue, in between our joints, as well as epithelial and neural tissues as well. So what does it do? It functions as a joint lubricant. So it's able to attract water. And so it works as a shock absorber and as a joint lubricant. But it also functions in wound healing, cell migration, ovulation, and granulation. And again, it's the key component and a key structural component of the extracellular matrix. So we'll move on to the next class of GAGs. These are known as chondroitin sulfate. So chondroitin sulfate is, as the name suggests, a sulfated glycosaminoglycan. What does that mean? That just means that we have different sulfate groups shown here that are found attached to different carbon or different oxygen carbons of our different monosaccharides. And so chondroitin sulfate is made up of mainly this disaccharide subunit of glucuronic acid and N-acetylgalactosamine. And typically, chondroitin sulfate can be sulfated at the 4 or 6 position, so here or here. And because it's sulfated at these different positions, chondroitin sulfate has a high negative charge, and it allows it to interact with many different growth factors and proteins, as well as other linker proteins that we'll talk a little bit more about in a second. So the difference between HA and CS and all these other GAGs is that all these different classes of GAGs are biosynthesized in the ER and the Golgi of the cell by a large group of biosynthetic enzymes that not only attach these individual sugars, but also there's a lot of sulfotransferase enzymes that will attach sulfate groups at these specific positions. And there's actually 4 different types of chondroitin sulfate, A, C, D, and E. And the difference between these different types are what type of sulfation patterns. And so what type of structure these different chondroitin sulfates have. And so chondroitin sulfate, like HA, is actually a chief component of the extracellular matrix, particularly in cartilage. And interestingly, chondroitin sulfate proteoglycans interact with hyaluronic acid. I'll show that in a later slide. So chondroitin sulfate, one of its main functions is it plays a key role as a shock absorber in our joints. And it also, because of its sulfation pattern, it also interacts with a lot of different important proteins and growth factors at the cell surface in the ECM as well. So we'll move to the next class of GAGs. This is very structurally related to chondroitin sulfate. It's actually also known as chondroitin sulfate B, but this is known as dermatan sulfate. So dermatan sulfate is, again, very structurally similar to CS, but the difference between CS and DS is that many of the glucuronic acid residues have been epimerized to iduronic acid. So if we go back here, you can see that this is the structure of glucuronic acid, and there's actually an enzyme or a couple enzymes that can epimerize glucuronic acid to iduronic acid. And this iduronic acid residue can actually be 2O-sulfated at certain positions. So the main structural difference between these two GAGs is that the presence of iduronic acid. And again, there's certain enzymes that epimerize glucuronic acid to iduronic acid to make a dermatan sulfate chain. So dermatan sulfate is mainly found in the skin, hence the name dermatan. And it's also found in blood vessels and plays a key role in blood coagulation. It's been implicated in cardiovascular disease, as well as in cancer, pathogen infection, and it also plays a role in wound repair as well. We'll move on to the next class of GAGs, and these are actually the class that I study in my laboratory. These are known as heparin sulfate or heparin. Both are very, very closely related. Heparin is a more highly sulfated form of heparin sulfate. And overall, in this class of GAGs, these are very highly sulfated. And because they can be sulfated in multiple different positions, these GAGs are involved in many important biological processes, such as cell signaling, stem cell development, cell-to-cell crosstalk, blood coagulation, angiogenesis, so the formation of new blood vessels, endocytosis of macromolecules. So very important for many different cellular processes, and as a result, these are ubiquitously expressed in all tissues. So heparin sulfate is essential for life as well. If you don't make heparin sulfate, you will not make it past the embryonic stage as well. So I mentioned a lot of the biological roles of heparin sulfate. Heparin, again, is a more highly sulfated form of heparin sulfate that's mainly produced in mast cells. So these are immune cells that will release heparin as well as histamines and other agents during inflammation. And we'll talk a little bit more about heparin because it's also one of the most highly utilized pharmaceuticals in the world, and it's used as an anticoagulant drug. So, but just keep in mind that heparin is also produced in mast cells as well. And so heparin sulfate is more ubiquitously expressed in all tissues. And looking at the structure, heparin sulfate also includes not only glucuronic acid, but it can also contain iduronic acid. So there's an epimerase enzyme that flips the glucuronic acid to become iduronic acid that can then be sulfated at the 2 position. And also the chains are made up of either N-acetylglucosamine or N-sulfated glucosamine. Again, these are very highly sulfated overall, and you get these sulfated domains that are very important for interacting with different growth factors and other proteins on the cell surface. And in the final class, we'll talk about keratan sulfate, or KS. So this is also a sulfated GAG, but unique to this class of GAGs is that this glycosaminoglycan can be attached to an asparagine or a threonine serine residue on its core proteoglycan protein. And this GAG is mainly made up of repeating N-acetylactosamine residues. So we have a galactose as well as an N-acetylglucosamine that can be sulfated at the 6th position. So there are specific enzymes that are involved in sulfating KS compared to, for instance, heparin sulfate or chondroitin sulfate. So just to keep in mind that there's unique enzymes that are involved in sulfating these different types of GAGs in our cells. So KS is found mainly in cartilage as well as the cornea of the eye, and also is found in bone. And it's important to not only, as a shock absorber in our joints. Again, these GAGs are hydrated molecules, but also KS interacts with many different proteins and ligands as well. So now that we've kind of gone over the structure of these different classes of GAGs, I'm kind of going to dive a little more in detail about these different, about the function and the biosynthesis of these different GAGs and what their role is in not only in our cells, but also in biomedicine. So we'll start with hyaluronic acid or hyaluronan. So HA is biosynthesized at the plasma membrane by a group of synthase enzymes known as the Has enzymes. And again, this is a unique biosynthetic mechanism compared to the other classes of glycosaminoglycans, because this GAG is biosynthesized right at the plasma membrane, and it is copolymerized with N-acetylglucosamine and glucuronic acid, repeating disaccharides, and it is not attached to our core protein. So the GAG chains exist as themselves, and they're pumped out into the extracellular space. And what's interesting about the biosynthesis of this glycan is that it's assembled at the reducing end. So the sugars are added right here by the Has synthases, and as they are added, the polysaccharide is pumped out into the extracellular space. And again, these are not attached to core proteins, and they exist outside of the cell. So what's interesting is once these hyaluronic acid chains are present outside of the cell, they not only form an architecture for the extracellular matrix. The question is, how do they do that? Well, they actually interact with different proteins, and these are known as HA binding proteins. And so there are many different proteins that interact with hyaluronic acid. And interestingly, many of the proteins that do interact with hyaluronic acid are actually other proteoglycans that they themselves have their own glycosaminoglycans, such as chondroitin sulfate on them. So here I'm showing a HA polysaccharide that's interacting, if we zoom in, with a linker protein that then connects it to another proteoglycan. So again, this is kind of how it is making up the architecture of the extracellular matrix. And just an interesting highlight for in medicine is that osteoarthritis, you see a degradation of these types of interactions of HA interacting with other extracellular matrix proteins as well. And so here, if we were going to zoom out and, for instance, look in the extracellular space from a chondrocyte, which, for instance, is in the growth plates of our bones, you could see that hyaluronan interacts with cell surface proteins as well as other proteins in the extracellular matrix, such as aggrecan, which is a proteoglycan that has chondroitin sulfate chains. Then it has these linker proteins that will interact and form kind of this mesh-like network of consisting of not only proteins, but also glycosaminoglycans as well. And so this is a major network that makes up our cartilage as well. So not only does HA function, as in our cells and in the extracellular matrix, in our tissues, but hyaluronic acid has also been utilized in biomedicine as well. So the thing that probably a lot of people will recognize is that a lot of cosmetic products actually contain hyaluronic acid. Again, this molecule can absorb water, and it's a great hydrating molecule, hence why it's included in a lot of different moisturizers. And besides that, it's also used in many other types of products, whether they're medical grade products or cosmetic products, for helping with wrinkling and aging and collagen stimulation, etc. So besides that, it's also been used, for instance, in drug delivery, where you can actually coat a drug with modified hyaluronic acid chains. They form these nanoparticles that will encode, for instance, drugs for drug delivery. It's also being investigated to be used as hydrogels, either to be used as a material for instance, wound healing, or also for growth of stem cells. So using it as a support for growing different cells or tissues in vitro or in vivo. So a lot of really a wide range of uses for hyaluronic acid in biomedicine. And again, this is still being explored and to be used in many different contexts. So we'll move on to the next class that we want to talk about, which is keratin sulfate. So again, the difference between keratin sulfate and, well, let's just say the difference between hyaluronic acid and all the other GAGs is that all the other GAG classes are biosynthesized in the ER and the Golgi of the cell. This is keratin sulfate, again, is made up mainly of this sulfated polyacetyl-lactosamine chain. And what makes keratin sulfate very unique from the other GAGs is that you can have these branched structures. Or for instance, the other chain is capped with a sialic acid and contains for instance, there's two different types of KSs, type 1 and type 2. And the difference between them is that KS1 is N-linked, so to an asparagine residue of its core proteoglycan. KS2 is typically O-linked. And because of that, the core glycosylation varies between these two types as well. But what is shared between them is this N-acetyl-lactosamine chain that can be sulfated. And I'm just showing two examples of two sulfotransferase enzymes that either add a 6-O sulfate group to the galactose or the N-acetyl-glucosamine. So this can be extended out as a long polysaccharide that can be sulfated, and this is where it interacts with different proteins and growth factors as well. And typically, sulfation is terminated by a terminal galactose. So you get sulfation of this terminal galactose, which stops the chain elongation. So again, the regulation of the length of these is dictated by the sulfation pattern as well, and by the specific enzymes that are involved in adding these sulfate groups. And I also wanted to highlight that KS biosynthesis is altered in different pathologies and during development as well. So, again, getting at the major point of why do we want to study the structure of GAGs? Well, it's because the structure is directly related to its function in our tissues and in our cells. So, for instance, if you change the sulfation pattern of certain GAGs, it has a profound impact on the function at the cell surface or in the extracellular matrix in terms of what these polysaccharides can interact with, or what signaling cascades are being activated. And so that's why it's developing new techniques to study the structure is so important for particularly glycosaminoglycans. So we'll move on to the next one. So this is chondroitin sulfate. So this is a non-sulfated, this is a sulfated glycosaminoglycan that can be sulfated at the 6 or 4 position mainly. And I'm highlighting here iduronic acid because if you remember from earlier, that if the chain contains iduronic acid, it's technically a dermatin sulfate chain. And so there's an epimerase that will make iduronic acid from glucuronic acid in these polysaccharides. Again, non-sulfated chondroitin sulfate is very rare in vertebrates. So most chondroitin sulfate and dermatin sulfate chains are sulfated. And depending on if they only have 4-O sulfation, for instance, they would be designated chondroitin sulfate A, and then B, C, D, E, so on and so forth. For instance, chondroitin sulfate E, which is very highly sulfated, especially with 6-O sulfate groups, is mainly found in the brain. It was originally discovered in invertebrates such as shrimp and other sea living creatures that also make these chondroitin sulfate chains. Just an interesting side note. And I guess, again, I wanted to highlight that there are multiple sulfotransferase enzymes that add these different sulfate groups to these different positions along the chain as well. And the main disaccharide subunit is a GalNAc with a glucuronic acid residue. And again, if it has the iduronic acid, it's typically labeled a dermatin sulfate chain. And we'll talk a little bit more about this later, but I have these arrows here pointing that one way that we can study the structure of these is to break down glycosaminoglycans into their disaccharide subunits. And then we can use different liquid chromatography and mass spec techniques to analyze the subunits that make up the entire chain. And again, we'll talk about this more in detail later. And these are the different types of chondroitin sulfates again, and that they can have different sulfation patterns. Again, if you only have 4 sulfate groups, it typically is going to be either of these chondroitin sulfate A or B. And also the different types of chondroitin sulfates or dermatan sulfate are found in different tissues as well. And so, not only does chondroitin sulfate act as a shock absorber in our joints, but chondroitin sulfate also interacts with many different factors, whether it be cytokines, growth factors, matrix degrading enzymes, different receptors on the cell surface. This chondroitin sulfate plays many different important roles in cell biology as well. Hence why it's studied in a lot of different contexts, such as wound healing, as well as in cancer. So a lot of cancers will upregulate chondroitin sulfate biosynthesis, for example. And just wanted to talk a little more about how chondroitin sulfate is biosynthesized, and I put HS here as well because we're going to follow up with this. But the biosynthesis of both of these GAGs is actually pretty similar. But here I'm showing that typically these are always O-linked and to a serine residue of the core protein, so the core protein glycan. And for chondroitin sulfate or heparin sulfate, biosynthesis is initiated by the attachment of a xylose residue to the serine residue of the core protein. And then 2 galactoses are added by specific enzymes. And then a glucuronic acid residue is added by this B3-GAT3 enzyme that adds the glucuronic acid. And this is what's known as the tetrasaccharide linker region. So this is common for chondroitin sulfate and heparin sulfate. I just wanted to highlight that here. But after this step, depending on what sugar is added next, it will go down these 2 pathways. Either where you get biosynthesis of chondroitin sulfate if you add an N-acetyl galactosamine, or if you add an N-acetyl glucosamine by EXTL3 is the enzyme that adds this, then you will biosynthesize a heparin sulfate chain. And I wanted to mention here that it's not completely understood what regulates this process, and this is part of the focus of my independent laboratory is we are trying to understand what regulates the biosynthesis of glycosaminoglycans in our cells. But in terms of the biosynthetic machinery, I just wanted to highlight here that depending on what sugar is placed next after this tetrasaccharide linker region, you will get biosynthesis of chondroitin or heparin sulfate. And so once you get addition of the repeating disaccharide subunit of N-acetyl galactosamine and glucuronic acid, you get elongation of the chain. And then these sulfotransferase enzymes will come and add sulfate groups along the chain. And these sulfate groups are very important for interacting with different ligands and different proteins as well. So that's a little background on chondroitin sulfate. Now we're going to move to heparin sulfate. So heparin sulfate proteoglycans, these are the set of known heparin sulfate proteoglycans. They can be transmembrane type proteins, such as the syndicans, GPI anchored proteins, such as the glycoscience, as well as these 4 different extracellular matrix proteins that are known to have heparin sulfate chains attached to them. And heparin sulfate plays an important role in many different biological processes, such as cell signaling, where they can interact and bind to different growth factors that then allow it to the growth factors to interact with the receptors on the cell surface. They can be involved in cell-to-cell crosstalk, where they can bind and present different ligands to receptors on adjacent cells, as well as their own cells, or their own cell surface. They're also involved in the endocytosis of different biomacromolecules or different lipids, such as lipoproteins, where these lipoproteins, for instance, can bind to heparin sulfate, and then the whole complex is endocytosed into the cell, where you can deliver different cargo into the cell, or it can be degraded through the endosome lysosome process. And heparan sulfate also is a key part of the basement membrane or extracellular matrix architecture, where it can interact with many different factors and other members of the extracellular matrix as well. So these are all the functions of heparan sulfate in normal important biological processes, but heparan sulfate has also been implicated in many different pathophysiological conditions as well. One of these main fields is the field of cancer, where heparan sulfate biosynthesis or proteoglycan expression can be upregulated in certain cancer types, and it also can be downregulated in certain types of tumors as well. And so heparan sulfate, because it's involved in binding growth factors and involved in cell growth and cell proliferation and angiogenesis and stem cell differentiation, you can imagine that cancer cells can hijack this system, upregulate heparan sulfate production, therefore enhance cell proliferation, metastasis, angiogenesis for the tumor, as well as differentiation to different lineages in different cancers as well. So besides cancer, also many different pathogens can hijack heparan sulfate on the cell surface. They bind to heparan sulfate, which then allows it to fuse the cell membrane and then infect our cells. So I'm showing here a picture of a herpes simplex virus that is known to bind to heparan sulfate on the cell surface. Interestingly, it binds to a specific sulfation pattern of heparan sulfate. Going back to what I said earlier about the major importance of the structure of these glycosaminoglycans really dictates its function in the cellular context. Also wanted to highlight here that recently last year, it was discovered that SARS-CoV-2, the virus that causes COVID-19, also requires binding to heparan sulfate. The spike protein binds to heparan sulfate on the cell surface, which allows it to helps it interact with the ACE2 receptor, which some of you may have heard of as well. So again, highlighting the importance of heparan sulfate for pathogen infection as well. So virtually most viruses that have been studied interact with heparan sulfate on the cell surface and utilize it in some way. So I'm showing HSV, HIV, CMV, many different flaviviruses. The list goes on, and even some bacteria also utilize attachment to heparan sulfate on the cell surface for infection. Moving on to neurodegenerative diseases, for instance, Alzheimer's and Parkinson's, heparan sulfate has been implicated in, for instance, the formation of plaques. So, for instance, amyloid beta is known to interact with heparan sulfate, and heparan sulfate is found to be a key part of these plaques that form in Alzheimer's patients, for instance. And then the last category that I'm highlighting here is that there are certain rare genetic disorders where there's either a defect in the biosynthesis of heparan sulfate or the breakdown or catabolism of heparan sulfate in the lysosome, for instance, of cells. And when you have a defect in either of these processes, it causes very gross phenotypes. I'm showing here that there's a rare genetic disorder called multiple hereditary exostoses, or MHE. This is a rare genetic disease that affects about 1 in 50,000 kids are born with this disorder. And it's a defect in the biosynthesis of heparan sulfate. And how this disease manifests is you get these bony outgrowths known as exostoses that are very painful, and they cause growth defects, chronic pain, and even a small percentage of these patients get cancer as well. And this is all due to a mutation in one enzyme that's involved in biosynthesizing heparan sulfate. And also there's a whole group of disorders known as MPS, or mucopolysaccharidoses disorders. These are lysosomal storage disorders where heparan sulfate cannot be catabolized in the lysosome of cells. As a result, many things affect these patients: mental retardation, growth defects. A lot of the patients would not even survive until they're 8 or 9 years old if it weren't for huge developments in different medications and treatments for these disorders as well. So again, just highlighting the importance of heparan sulfate not only in normal biology, but also in different pathologies as well. Therefore, it's really important to understand not only how these sugars are biosynthesized, but also, how are they regulated in our different cells? And we'll talk a little bit more about that shortly. So here I just wanted to highlight, we already kind of talked about how chondroitin sulfate is biosynthesized. Heparan sulfate, again, the first 4 sugars, which make up the tetrasaccharide linkage region, are shared between chondroitin and heparan sulfate. But also in heparan sulfate, we get attachment of the xylose residue, which can be phosphorylated. And I mentioned this was more recently discovered, that this phosphorylation event can affect the biosynthesis of these different GAGs as well, chondroitin and heparan sulfate. So we get the tetrasaccharide linker region. Then again, if we get the addition of N-acetylglucosamine, we are now going to be biosynthesizing a heparan sulfate polysaccharide. And then there's 2 enzymes that form a copolymerase complex, EXT1 and EXT2. So these copolymerize the chain with N-acetylglucosamine and glucuronic acid. And once you get polymerization of the chain, then we have a large family of sulfotransferase enzymes that will come and add a sulfate group to different positions along the chain. And we also have this epimerase enzyme that I mentioned earlier that epimerizes glucuronic acid to iduronic acid that can then be sulfated at the 2 position as well. So, once we get sulfation of heparan sulfate, if we kind of zoom out on the whole chain, this is what heparan sulfate actually looks like, is that that we have these sulfated domains, and then we have non-sulfated or very lightly sulfated domains, known as acetylated domains. And these sulfated domains are very important for binding different growth factors and other ligands on the cell surface. So for instance, antithrombin, which is a serine protease inhibitor, this factor is involved in the blood coagulation cascade. So it's an anticoagulant that is not as active as when it interacts with heparan sulfate and the more highly sulfated form of heparan sulfate known as heparin. So what happens is antithrombin is able to bind to this specific pentasaccharide subunit of heparan sulfate. So it recognizes this specific subunit that's sulfated at these different positions. And I want to highlight that this 3-O-sulfate group is very important for binding of antithrombin. Interestingly, this 3-O-sulfation is relatively rare modification of heparan sulfate, but it's very, very important for binding to antithrombin. And once antithrombin binds to heparan sulfate, this causes a conformational change in antithrombin, which then enhances its activity 4,000-fold, around 4,000-fold, to be able to inhibit blood coagulation factors. So as a result, heparan sulfate and heparin, which is an anticoagulant drug, how it works is it binds to antithrombin in our bloodstream, activates antithrombin, and inhibits the blood coagulation cascade. So it prevents blood clotting. So this is typically used during surgery, but typically in normal, homeostasis in our tissue, heparan sulfate on the cell surface, so for instance, our endothelial cells will interact with antithrombin and regulate the blood coagulation cascade. So this is an example of a protein that binds very specifically to heparan sulfate and recognizes a specific sequence. There's also other factors such as fibroblast growth factors, as well as other growth factors that instead of binding to specific sequences of heparan sulfate, they more recognize certain sulfation patterns. So for instance, FGF2, which is one fibroblast growth factor, interacts with N and 2 sulfation patterns and N and 2 sulfation sulfated residues of heparan sulfate. And also FGF receptors on the cell surface will also interact with heparan sulfate as well. And interestingly, heparan sulfate has been found to help form a complex between all of these different proteins. And this is essential for cell signaling and development, for instance, during stem cell differentiation, as well as normal cellular growth and proliferation. There's other factors such as chemokines that, for instance, can bind across these sulfated and non-sulfated domains as well. So in this case, heparin sulfate can act as a sink for binding different factors on the cell surface and preventing them, for instance, for activating different signaling cascades downstream or in other cell types. And so heparin sulfate plays a role in not only signaling, but also can bind and hold on to different factors that then regulates signaling cascades and other pathways for normal cell biology. And so while here I'm showing all these enzymes that over the last many decades, all these enzymes were discovered to be involved in the biosynthesis of heparin sulfate. And while we know how the biosynthesis typically occurs for heparin sulfate as well as other types of glycosaminoglycans, what's not really understood is what regulates the production of not only heparin sulfate, but other glycosaminoglycans. So why is there such a diversity in structure across different tissues, for instance, or in disease states such as cancer versus normal tissue? So here I'm just showing from some published data. For instance, if you pull out all the different tissues of a mouse, for instance, and we look at heparin sulfate levels in these different tissues, you can see it varies pretty widely between different tissues. And again, it's not fully understood at this point what regulates these differences in heparin sulfate amounts. And on the right here, I'm showing that these are techniques that I'll talk a little bit more in detail later, but where we can actually break down these different GAGs, for instance, heparin sulfate, down to the disaccharide subunits. Then we can use liquid chromatography and mass spectrometry to analyze what disaccharide subunits are present in our sample. For instance, if we isolate GAGs from different tissues. And we can actually use standards where there are known structures, and we can match the standards to different disaccharides that we find from our different samples. And from this, we can kind of piece together a composition of our different GAG chains. So we can say that for instance, the heparin sulfate chains from a bladder carcinoma contains this percentage of this particular disaccharide, which in this case is a non-sulfated disaccharide. But you can see we can look at all these different disaccharides with different sulfation patterns, and in effect, we can identify the composition of our different GAG chains in different tissues. And this tells us structurally what the diversity looks like, for instance, in different tissues or in different diseases or different cancer types or tissue types as well. And again, I'll talk a little bit more about this later, but I just wanted to highlight here that the diversity and the structure of these different glycosaminoglycans is very diverse between our different tissues. And the structure of these different GAGs has a direct effect on its function in vivo. So I just wanted to highlight here, this is kind of the role, or the kind of one of the goals of my research lab, is that we're trying to understand what regulates the production of glycosaminoglycans, and currently our focus is on heparin sulfate due to its wide range of functions, not only in normal biology but also in different diseases. And so you can imagine before any of these glycosaminoglycans are presented on the cell surface, there's a lot of different steps along the pathway that could be regulated. There could be transcriptional or translational regulators. So it's already known that transcription and epigenetic factors regulate gene expression across the genome. And it's not well known what regulates the expression of these different enzymes. So for instance, some of these enzymes are only expressed in certain tissues. Why is that? We don't know, but we can assume that there's some kind of transcriptional or translational machinery that is turning off or on the expression of these different genes. And so for instance, if you don't express this HS2ST enzyme, you will not get 2-O-sulfation of your heparin sulfate chain. And this can have downstream impacts when it's presented on the cell surface with what growth factors or other proteins it interacts with, and then as a result, which signaling cascades are being activated in certain cell types or tissue types. And so we're using different techniques to look across the whole genome, and for instance, using genome-wide CRISPR-Cas9 screening, where we can, in effect, remove or knock out every single gene in the human genome individually in cells, for instance, in human cancer cells. And we can ask the question, when I knock out or remove a certain gene X, Y, or Z, how does that affect the biosynthesis of heparan sulfate? And so we're developing methods where we can look across the whole genome for factors that might be regulating this biosynthetic process. And so we've already identified certain transcription and epigenetic factors that regulate the expression of specific enzymes involved in heparan sulfate production. And so, of course, the end goal is to identify ways that we can utilize these findings to help patients, for instance, that do not make enough heparan sulfate. Can we identify a drug target where we can increase heparan sulfate levels? Or if, for instance, you can't break down heparan sulfate levels, can we identify targets or drugs that will lower heparan sulfate production as a substrate reduction therapy, for instance, for lysosomal storage disorders? And we also use many different bioinformatic tools and other genomic tools to look across the genome and look in our DNA and ask the question, are there answers hiding in our own DNA? So are there transcription factor binding motifs that are enriched across these different enzymes? What decides if a proteoglycan is going to have a heparin or a chondroitin sulfate chain? Are there certain factors, for instance, in the ER and the Golgi that might regulate how all these enzymes are coming together to form complexes that will then, in effect, biosynthesize these chains? So I just wanted to give a quick highlight here before we move on that, these are still a lot of outlying questions in glycobiology. And I'm highlighting GAGs, obviously, in this talk, but this expands to other types of glycans as well. What regulates their production and their function in cells. And again, the major reason why we're interested in structurally analyzing these different GAG chains is that the structure dictates what proteins or factors these chains are able to interact with in vivo. And so not only do we know all about these enzymes that are involved in biosynthesizing these chains, but we're also asking the question of what proteins interact with these GAGs and how does the structure impact the interaction with different proteins as well. So, the analytical service core here at the CCRC that's running this whole program, this is their specialty is how do we analyze the structure of glycans and then we can relate that to its function. And so our lab, as well as many other labs here at the CCRC, collaborate with the analytical facility here and help us to hopefully get a clear picture and how what is the structure of these different glycans in different biological contexts and different pathological contexts as well. And again, these I'm showing it here as a cartoon model, but in reality, these different proteins are interacting in a 3D manner with the different GAG chains. So this is the complex I mentioned earlier, where we have a heparin or heparan sulfate molecule that is helping pull together a growth factor and its receptor in what's known as a ternary complex. And this is what typically happens on the cell surface. And this is essential for the growth factor to bind to its receptor, which then can signal to downstream signaling pathways in the cell. I just mainly am highlighting different ways that these glycans can interact with different monomeric or dimeric or oligomeric forms of proteins. And so not only is heparan sulfate involved in forming a co-complex, but it's also involved in oligomerizing certain proteins as well. So here I'm showing RAGE, which is the receptor for advanced glycation end products. This is a inflammatory receptor that is involved in obviously in inflammation, but it's also implicated in cancer and diabetes and many other disorders as well. And heparan sulfate was found to be essential for oligomerizing RAGE, so it could be its functional form where it can then interact with other types of ligands and affect downstream signaling as well. And then you can also have the interaction where it's more on the outside of a protein, for instance, where you get where it can also help proteins dimerize as well. And, as I mentioned earlier, heparin sulfate or heparin, the more highly sulfated form, can interact with antithrombin, which causes a conformational change in the protein as well. So just to highlight different types of GAG protein complexes and how the interaction can have a profound impact on protein function. And here I just wanted to highlight a little more in detail. Again, I mentioned earlier that heparin is a very potent anticoagulant drug. So this is a drug that's, it's $1 billion industry. It's used widely during surgeries to prevent blood clots. It's also can be used in many other aspects as well, and different forms of heparin are also used in outpatient care as well to prevent blood clotting. Again, heparin is a more highly sulfated form of heparin sulfate. So it has a very similar structure. It just has very high sulfation levels. And I wanted to highlight that this relatively rare modification on heparin sulfate, the 3-O-sulfation group, heparin has a higher than average amount of 3-O-sulfation. And interestingly, 3-O-sulfation, even though it's thought to be the rarest modification, there are 7 different enzymes that are involved in adding the 3-O-sulfation group to different residues along the heparin sulfate or heparin chain. Very interesting that there's this many isoforms, and it's not completely understood why that is. There's still a big hurdle in understanding the structure of and identifying 3-O-sulfated structures in heparin sulfate as well. So this is still a big area of research as well. But I'm highlighting it here because this 3-O-sulfation group is essential for heparin's function as an anticoagulant drug. When the 3-O-sulfate group is present in this pentasaccharide subunit, it can bind to antithrombin, which causes a conformational change and enhances antithrombin's activity to inhibit the blood coagulation cascade. Hence why it's a potent anticoagulant drug that's used all around the world. And you might ask, where is heparin isolated from? Well, it's actually a natural product. And so this drug that is so widely used is actually isolated from pig intestines in China mainly. And it's also isolated from bovine lung, but this type of heparin is mainly used in Europe. So the most widely utilized heparin is mainly from pig intestines and mainly produced in China. And again, heparin, this more highly sulfated form of heparin sulfate, is mainly produced in mast cells. And it's typically stored in these granules along with histamines and other inflammatory factors. And during infection or inflammatory response, it's released out of these mast cells and is able to obviously interact with different proteins and ligands in that context. But in terms of a natural product, the porcine mucosa is scraped out of the pig intestines, and then heparin is purified from this. And back in 2008, there was an interesting case of a very sad case that a lot of people died from a contamination in the heparin stock. It ended up being an oversulfated form of chondroitin sulfate that was the contaminant that was found in this heparin stock. So I'm just highlighting this because there's still a lot of interest in, can we move away from isolating heparin as a natural product? Could we bioengineer cells, for instance, in a laboratory in a highly controlled setting to produce anticoagulant heparin for a drug? So this is something that a lot of groups are looking at. And can we bioengineer cells to produce highly sulfated, 3-O-sulfated heparin for biomedicine applications? Yes. And to dive a little bit more detail, I mentioned that heparin binds to antithrombin and causes this conformational change. Once it causes the conformational change, now antithrombin can go inhibit different blood coagulation factors, mainly factor 10A and factor 2A, which are involved in the blood coagulation cascade. And I wanted to highlight here that one of the clinical assays that's used in hospitals, so when you dose a patient with heparin, you need to monitor heparin levels in the bloodstream. So one of the ways that they monitor that is looking at factor 10A activity. And in the lab here, that's what we can do as well to monitor the activity of heparin and heparin sulfate in the context of blood coagulation as well. I think that's all I wanted to highlight here. So next, let's move on to, okay, we now understand the biological function of GAGs in different contexts, depending on what class of GAGs we're talking about. We know how they're biosynthesized in our cells. We don't really understand how they're regulated. But what we can do is there have been many methods that have been developed to isolate and purify and structurally analyze glycosaminoglycans. And again, one of the big hurdles in analyzing GAGs is how complex their structures are, right? If we go to hyaluronic acid, that only contains these two non-sulfated sugar subunits, and it's just a disaccharide repeating unit of that. But keep in mind that as we move to the other classes of GAGs, you get these various sulfation patterns that are, again, very important for the biological function. So many different methods that have been developed and are continuing to be developed to try to analyze, the structure of glycosaminoglycans. I'm just highlighting a few different tools that I use in my lab that are also, utilized in the Analytical Service Corps here at the CCRC. So the main thing is you first need to take your cells or tissue of interest, and then we use different chromatography strategies to isolate these negatively charged molecules. So we use anion exchange chromatography or size exclusion chromatography. And then we're able to either analyze the whole chains, or we can break down these chains into their disaccharide subunits, and for instance, do a disaccharide analysis, where we can look at the presence and the amount of different disaccharide subunits of glycosaminoglycans. Additionally, we can use different affinity chromatography techniques, where we can attach the glycosaminoglycan itself to the resin and then pass different proteins over this. What this allows us to do is to ask which proteins are able to interact with different types of glycosaminoglycans. We can also, isolate different structures of glycosaminoglycans, append them to different resins, and then ask how that affects the binding and interaction with different proteins as well. You can also flip it around, where we can make different affinity columns where we attach different proteins, and we can put different glycosaminoglycans with different structures over these columns. And we can ask, which different types of GAGs bind to our protein of interest. We can also use size exclusion chromatography. Again, this is where we can look at the size of the intact glycosaminoglycans in different tissue types or in disease states versus normal tissue. So for instance, in cancer, or what I'm showing here, if you have a rare genetic disorder where there's a defect in the biosynthesis of heparin sulfate, you can get shortening of the heparin sulfate chains, and we can use size exclusion chromatography to measure the overall size of the chains produced by a certain cell type. And finally, we can also use surface plasmon resonance or flow cytometry to look, for instance, at cell surface binding. So we can incubate different growth factors that are known to bind to, for instance, heparin sulfate or other glycosaminoglycans. And we can look at, for instance, in a normal cell versus a mutated cell, whether if you knock out one of the sulfotransferase enzymes, or for instance, in a cancer cell versus its relative normal cell, we can look at the binding of different growth factors on the cell surface. Or we can take this to like an SPR type application where we look at the direct interaction between a factor and a protein and the glycosaminoglycan chain itself. And again, I wanted to highlight that one of the big struggles still in analyzing glycosaminoglycans is understanding what the entire chain structurally looks like. And again, these techniques are being developed, and a lot of labs actually here at UGA are kind of pioneering these techniques. And we the one thing that we do have down really well is we can degrade these GAGs into their disaccharide or oligosaccharide subunits using different GAG degrading enzymes known as lyases or hydrolases. So these are two different types of enzymes that, for instance, hyaluronidases will cleave right here at this beta-1,4 linkage between the N-acetylglucosamine and glucuronic acid. So you can break down HA to its disaccharide subunits, and you can analyze. You can either remove HA in certain contexts and ask how that impacts biology. We could also do size exclusion chromatography as well to look at the size of different chains in different contexts as well. Chondroitinases are a group of enzymes that cleave at different positions of chondroitin or dermatan sulfate. So we typically can use individual enzymes, or we can mix different enzymes together. Again, these digest different types of chondroitin sulfate. So chondroitinase A will cleave a chain that has mostly 4-O-sulfation, for instance. And then chondroitin sulfate B will cleave more of a dermatan sulfate type chain. And so we can use these different enzymes to break down CS and DS chains, for instance, to their disaccharide subunits. And then we can use liquid chromatography to separate these different types of disaccharides. And again, they resolve pretty nicely because the different disaccharide subunits can have different sulfation patterns, as well as different monosaccharide subunits that affect how the chromatography goes for these different disaccharides. And then we can use mass spectrometry as well to not only identify what disaccharides are present, but we can also use that to absolutely quantify the amount of these different disaccharides as well, using standards, for instance, that are isotopically labeled is one application. We'll talk a little bit more about that shortly. And again, just highlighting, we also have similar enzymes called heparinases that cleave heparan sulfate at specific positions. What's really nice about these different enzymes is that some of these enzymes will only cleave at non-sulfated domains of the chain. And then some, like heparinase 3, will cleave at very highly sulfated positions of the chain. And so we can use a cocktail of enzymes to cleave heparan sulfate down to its disaccharide subunits. Or you can use individual enzymes, for instance, if you want to look at just the domain structures that are present. So if you use an enzyme that cleaves at the non-sulfated domain only, you'll end up with large oligosaccharide domains that will either be sulfated or non-sulfated, depending on which enzymes you utilize. And John Amster and Franklin Leach's groups here at UGA have been really pioneering work on analyzing these larger domain structures, as well as looking at the entire chains, for instance, for dermatan and chondroitin sulfate. And I'll share some of those references later if you're interested in learning how they do that. So using different mass spec techniques to fragment the chains in a certain way where you can actually piece together what the sequence of the chain is as well. And finally, keratinases are another group of enzymes. Again, there's different enzymes that will cleave different sites of keratin sulfate as well. And so these are really useful tools to help us analyze the structure of these different GAG chains. And also want to mention that there are other tools and chemical agents that are used to also cleave these chains. So for instance, nitrous acid is a chemical that is used to cleave, for instance, heparan sulfate down to its oligosaccharide and disaccharide subunits. And another tool that can be used for this as well, I didn't prepare a slide for it, but I wanted to mention that because most of these GAGs are highly sulfated, you can actually feed cells a radio labeled sulfate. So for instance, sulfur 35 sulfate that's commercially available, you can feed that to the cells, and they will actually incorporate radioactive sulfur into their GAG chains. And you can actually utilize this to quantify GAG chains. We can purify these radio-labeled GAG chains, and then we can structurally analyze them more easily using that method as well. So that's kind of an orthogonal method that can be used along with many of the liquid chromatography and mass spectrometry techniques that have been developed as well. And I kind of already mentioned this, but the big hurdle still in analyzing glycosaminoglycans is how do we sequence the entire chain? So far, we could be really good at breaking these down into their oligosaccharide and disaccharide subunits, and this is what's known as bottom-up sequencing, where we can actually we can piece together the subunits back and we can kind of get an idea of what the chain may look like in terms of its sequence. So there's different groups in Sweden as well as here in the States that are developing tools and I just wanted to highlight this really cool technique where you can actually feed the cells a xyloside that is labeled. And what this is this xylose will actually be utilized to start producing a chondroitin sulfate or heparin sulfate chain attached to this chemical. And what they can do is they can actually purify this and use different mass spectrometry techniques to analyze the GAG chain that was produced on their xyloside that they fed the cells. So it's a really cool chemical biology type technique, and where they can then use different chondroitinase enzymes for chondroitin sulfate, and they can cleave the GAG chains down to their subunits, and then they can piece back together by fragmenting these different chains with different mass spec techniques. They can piece back together a potential sequence for these different GAGs. This is known as bottom-up sequencing. But there's also been many advances in what's called top-down sequencing. This is where you take the whole chain or a large oligosaccharide and you use different mass spec techniques to fragment the chains. And then you can look at based on the fragmentation patterns, you can piece back together what monosaccharide subunits and sulfated subunits were attached to each other in terms of understanding the sequencing. And so this type of technique was utilized to sequence a chondroitin sulfate chain from bicunin, sorry. And so this is a proteoglycan that contains chondroitin sulfate chains. And also was utilized to sequence a decarin chain, which contains dermatin sulfate, I think, as well as chondroitin sulfate as well. And then finally, something that's actually very new and was only just starting to get published on. People have thought about this for a long time, but actually starting to utilize this. Bob Linhart's group up in New York, as well as many other groups, are starting to use solid state nanopores, which were originally developed to help sequence DNA and RNA. Now because GAGs are also negatively charged, and each sugar subunit can potentially have a different sulfation pattern as well as a different type of sugar. The idea is, can we detect different types of sugars as they pass through an electric current? And then we can, the idea is, can we use this to actually sequence the GAG chain one sugar at a time? So this is still in development, but there's been some recent advances that are getting us closer and closer to be able to, hopefully one day we can just sequence GAG chains in their native state. But yeah, this is a very exciting area of research in terms of the structural analysis of GAGs. So, that's on the structural side, but we could also look at how the structure impacts the function of GAGs by, for instance, using chemoenzymatic or synthetic organic chemistry to synthesize glycosaminoglycan oligosaccharides. And we can actually attach these to microarrays. And we can then ask different questions like, how does a certain growth factor or protein interact with different types of GAGs and different types of oligosaccharides? So this is a great orthogonal method where we can understand how the structure impacts the function. And also wanted to point out that synthesizing these GAG standards is not trivial. And so Guillaume Moon's group here at the CCRC, Jian Liu is another big lab that's involved in synthesizing, for instance, heparin sulfate oligosaccharides for the use in microarrays. as well as standards for mass spectrometry techniques, and also utilizing them as potential drugs as well to block certain heparan sulfate or GAG protein interactions as well. And the last little section, I just wanted to highlight that, as I mentioned before, there's also many lysosomal storage disorders where there are defects in the catabolism of glycosaminoglycans. So typically, especially cell surface GAGs are presented attached to their core proteoglycan protein on the cell surface. And these are typically endocytosed and recycled, or and where they can also be degraded through the endosome-lysosome system. And there's a whole group of enzymes that are involved in, and these are all glycosidase enzymes or sulfatase enzymes that are involved in breaking down the heparan sulfate or glycosaminoglycan chains in the lysosome at these low pHs. And interestingly, how these GAGs are digested or catabolized is exactly the opposite of how they're biosynthesized. So again, everything except for hyaluronic acid is typically added from the non-reducing end. So meaning that these sugars are added in this method, while hyaluronic acid, as it's pumped out of the cell, it's added on the reducing end. So I just wanted to review that really quick. But for most GAGs, these sugars are added on the non-reducing end. And interestingly, when these are broken down in the lysosome, the enzymes come and cleave each sugar and sulfate group starting from the non-reducing end towards the reducing end. And in effect, what you get is, sulfate groups of different monosaccharide subunits that can then be recycled or broken down further for further use in the cell. So these lysosomal storage disorder, and particularly a group of them are known as mucopolysaccharidoses or MPS disorders. These disorders are where the patient has a genetic mutation in one of the enzymes involved in degrading GAGs in the lysosome. And interestingly, if you are having a mutation and typically these are all autosomal recessive, so you have a homozygous mutation in one of these enzymes, all you need is a mutation in one of these enzymes and you get a large buildup of heparan sulfate or other GAGs in the lysosome of the cell. Because again, all the enzymes are necessary to stepwise break down these GAG chains in the lysosome. So if you're missing one of them, you get a buildup of certain oligosaccharides. So for instance, if you are missing the enzyme that removes this 2O-sulfate group, in effect, what you'll get in the lysosome is a large buildup of your heparan sulfate chains in this case, where the non-reducing end they will all mostly have this non-reducing end sugar with the 2O-sulfate group because you're missing the enzyme that removes this. And this might help a little more visual way of showing you how this is stepwise broken down. One sulfate group, one sugar at a time. So these are the different enzymes that are involved in removing, for instance, the 2O-sulfate group that I just mentioned. So iduronate 2-sulfatase removes this 2O-sulfate group. After this, then you get iduronidase, which removes this iduronic acid. And then so on and so forth. You have these different enzymes that are involved in removing these different sulfate groups. And it works in a processive fashion for degrading these GAGs. You can see that there are a different set of enzymes for different types of GAGs. So chondroitin sulfate has its own specific enzymes that remove, the 4O-sulfate group that is not found in heparan sulfate, for instance. And so, I'm introducing this here in this course because one big, application to understanding the structure of GAGs is that can we utilize tools to analyze the structure of GAGs, for instance, to identify biomarkers for different diseases such as MPS disorders? Or can we utilize, if a certain treatment is working, for instance, if we wanted to restore expression for instance, if you have a homozygous mutation in this enzyme, you get a buildup of heparan sulfate in your lysosomes and it causes gross phenotypes. And most of these patients would not make it past 8 or 9 years old if it wasn't for these these huge strides in, for instance, reintroducing the missing enzyme. So there's different ways of tackling these types of disorders where if you have a mutation in this enzyme, you can now get infused with, for instance, iduronidase enzyme. This was developed by Biomedicine, which is a rare disease company. So these patients get, for instance, weekly infusions of the missing enzyme known as enzyme replacement therapy. And so, other aspects, gene therapy is kind of a newer thing that has potential for, can we just, use gene therapy to train cells to continually make this missing enzyme? Or what I mentioned a little bit earlier is, can we use substrate reduction therapy to, for instance, reduce heparan sulfate biosynthesis to kind of lessen the load on lysosomes of these patients, for instance. So if you make less heparan sulfate, you might get less buildup of these non-degraded GAGs in the lysosome. And so here I'm highlighting all the different disorders, that the names that have been designated for these patients with different enzymes and missing enzymes. And again, this is important. If we can understand the structure, we can then understand not only the function of GAGs, but also certain biomarkers that might be present. So you can imagine if you do not make this enzyme, you're going to get a buildup of heparan sulfate chains that have this non-reducing end sugar or disaccharide. And so what we can do is we can utilize mass spectrometry techniques to recognize and detect these GAG-based biomarkers that are present in these patients. And so these types of tools can be used to detect these different biomarker monosaccharide or disaccharides in, for instance, the urine of patients or in the bloodstream. And so these are easy ways that we can isolate GAGs from biological samples and, for instance, look for biomarkers using liquid chromatography mass spectrometry techniques. So one of the things that's made this all available is that many people have developed standards where we can use different lyases to digest heparan sulfate or different GAG chains down to their disaccharide subunits. And then we can use these as standards by tagging these standards, for instance, with isotopically labeled molecules such as aniline. And this allows us to detect our standards compared to our patient disaccharides as well. And so when you run these out, for instance, on a liquid chromatography system, you can see that different disaccharide subunits of, for instance, heparan sulfate separated out really nicely using this chromatography system. And what we can do is we can spike in standards, for instance, that contain, instead of a normal aniline, they contain C13 aniline. So where all the carbons are isotopically labeled. And so what that allows us to do is at the same retention time, you'll have your standard that will have a higher mass shift than the, for instance, patient sample or biological sample that you're interested in. And so if you spike in a known amount of standard, you can actually absolutely quantify how much of a different disaccharide or a different biomarker is present in your sample of interest. And so I'm just showing you here that, for instance, if we make, for instance, we look at a mouse model of a lysosomal storage disorder where they're missing an enzyme that's, for instance, involved in removing this iduronic acid, you get a buildup of this disaccharide, not only in human patients, but also in these mice. And what we can do is we can use standards to detect how much of this disaccharide is present in our sample. And as you can see, for instance, in the mutant mouse, you get this huge buildup in this standard. So these types of tools are actually being utilized in the clinic to detect and to measure, what effect, that certain genetic mutations have on these patients as well. So I think I'm going to stop there. So there were some questions in the chat, but yeah, if there's any questions, I'd be more than happy. And thank you all for attending. And again, I'd be happy to answer, more than happy to answer your questions that you have.
So there are a couple of questions. I'll see if people have additional ones. So there were a couple of statements appreciating the focus on, the emerging fields for recombinant heparins.
Yes.
So yeah, totally agree on that one.
Cool.
So one question that was kind of discussed back and forth was whether or not you could make heparin sulfate affinity columns that are specific for specific sulfation patterns.
Yes, no, that's a great question. And yes, you can do that. So I'll just mention first that, for instance, heparin affinity columns, so heparin cephalose is commercially available. So that's a really nice tool if you need to purify, for instance, factors that bind to heparin sulfate and everything. So heparin cephalose, is commercially available. But yes, we can also attach different types of heparin sulfate, either isolated from biological materials such as tissues or cells, or recombinant material as well. So you can do it through different chemistries. So there's different, for instance, cephalose beads that you can purchase, or you can, for instance, use peptide coupling to attach different heparin sulfates, for instance, different gags, or you can take advantage of the amine groups that are in heparin to then do NHS ester chemistry. So where you can actually attach the gags through different amines that are present on the gags and bind that to the NHS ester. But yeah, so people have used this for many different applications. For instance, my previous mentor, Jeff Esco's group at UCSD, has utilized this to look more in detail about 3-O-sulfated heparin sulfate. So we can actually engineer cell lines. And I think this kind of will answer another question I saw in the chat, is that you can engineer cell lines to make different types of sulfated heparin sulfates. So if you knock out all the 3-O-sulfotransferases, for instance, these chains will not have 3-O-sulfate groups. Or if you knock out 2-O-sulfate groups or 6-O-sulfation enzymes, you will remove those types of sulfate groups from the chain. And then those cells or whatever will make those types of heparin sulfates. We can isolate and purify those and then attach them to affinity columns, for instance, or as I mentioned, to microarrays. That's another way. And some groups are even, synthetically producing heparin sulfates or using polymer chemistry as well. Camille Godul's lab is pioneering a lot of this type of chemistry, where you can actually make synthetic proteoglycans. So they use proteins where they add different heparin sulfates, and then you can actually insert these into the membrane of cells. So different ways that we can take heparin sulfates or synthetically produced heparin sulfates and then use them in this way, whether it's affinity columns or other applications as well. Hopefully that makes sense. I think someone was asking about a paper. Let's see, for eluting from heparin columns. Yeah, I'd be more than happy to send that. If you don't mind, maybe getting in touch with me through email. Is my email available, Stephanie, to everyone?
I can make it I can pop it up really quick.
Okay, great. Or I can do that, I guess, as well. Yeah, feel free to reach out. So yeah, I'm just, the paper I just highlighted is one from my previous group, but I've also utilized that as well. And so I can send you some of the papers that we've published as well to engineer. And, part of coming back to the bioengineering heparin, that's been a great effort of my lab and others, can we identify, factors that can help us produce this highly sulfated 3-O-sulfated heparin sulfate to act as a bioengineered version of natural product heparin as well. Any other questions?
They're pretty quiet.
Yeah, that's okay. But yeah, feel free to reach out if there's anything I can help with or any questions that I can have answered.
It was a great talk. Thank you so much.
Oh yeah, no, thank you for having me. And thank you all for listening, and hope everyone has a great rest of the day.
All right.
All right.
All right.
Thanks, Stephanie. Bye.
All right, guys, we'll see you all bright and early tomorrow morning. Topics tomorrow include NMR combined with some LCC and some great talks on the mass spectrometry of GAGs. Have a great evening.